Okay, so we knew this was possible. We've all seen the movies, read the papers, know the horror stories. I honestly am freaked out about being here and honestly didn't think I was going to win the contest to even be here. What makes you think that what happened the first park won't happen now?
Seriously, like, I'm all about action and adventure, and what I've seen today, I mean, it's alright, I guess it's kind of incredible, but I don't want to be killed. I'm not trying to die, so, you know, and like, I just figured there was like robots and not really dinosaurs. I thought they were like fake, you know.
I'm fully aware of my grandfather's legacy, trust me. I had to deal with that throughout my entire childhood. We studied what went wrong with the first park, and we made sure this time that none of that could possibly happen. We've upgraded the fences, the security systems. Anyways, what we really discovered, the problem was that the dinosaurs were able to morph sexually, going from females to males and mating with one another. So what we did this time, instead of splicing in frog DNA, is going with reptiles DNA or avian DNA. And we've actually been able to create much closer than my grandfather the actual original animals from the Paleozoic era.
Scientist, this is Dr. Wu, no relation to Henry Wu.
Hello.
She will start to fill you in on how we extracted the DNA from the amber. Dr. Wu.
Thanks.
As you may know, DNA, or deoxyribonucleic acid, is the carrier of genetic information. It is found in every living cell. The structure of DNA was discovered by James Watson, Francis Crick, and Rosalind Franklin in 1953. The men received the Nobel Peace Prize for their efforts, while all of Rosalind's work went overlooked because of her death of cancer. In the case of DNA, it is twisted in a right-handed manner and contains major and minor grooves. To give an example of the size of DNA, for every 10 bases or nucleotides of DNA is equal to about 34 angstroms, which is very, very small. For example, there are 3 trillion angstroms in just 1 foot.
We already know this. It's made up of a sugar phosphate backbone and nucleotides. The sugar phosphate backbone of each strand of the DNA runs anti-parallel to each other in the 5' to 3' direction. It has to do with the location of the phosphate group on the 5 end and the hydroxyls on the 3 end.
Yes, but do you know the differences between the sugars? The DNA is missing an oxygen group on the 2' carbon. A nucleotide is a part of DNA that carries the information needed to make protein.
The specific order of the nucleotides determines the differences in heredity.
There are 2 types of nucleotides, the pyrimidines and the purines. The pyrimidines are composed of a single carbon and a nitrogen ring and consist of cytosine, thymine, and uracil. However, uracil is only found in RNA. The purines are composed of a double ring structure. They are guanine and adenine. In DNA, adenine bonds to thymine, forming 2 hydrogen bonds between them. Guanine binds to cytosine using 3 hydrogen bonds, making the GC bond stronger than the AT bond. This gets into how we made the dinosaurs. A set of nucleotides or gene acts as a code for the transcription of RNA, which will be translated into proteins. This is called the central dogma of biology. The central dogma theory is the process of transcribing the DNA into RNA and then translating the RNA into proteins. Transcription occurs in the nucleus, and translation occurs in the cytosol of the cell. As I overheard Mr. Hammond explain to you, we no longer utilize mosquito to extract DNA. It is too degraded. Instead, we have found skin cells and certain types of amber from a tree that existed worldwide. These skin samples are infinitely more reliable than blood from mosquito. As a
You can
result,
survive on it. Tastes like shit.
As a result, few of our species require massive splicing to complete each DNA chromosome. DNA does this thing called alternate splicing, so lots of different proteins can be made, which are needed for every organism to survive. Each gene is made up of introns and extrons. During splicing, all the introns are removed, and certain extrons are either removed or kept and translated into RNA, giving rise to variation
Dr. Wu.
in proteins. Yes.
What do you get when you cross a T-Rex with a pig?
I have no idea. What do you get?
Jurassic Park.
Good one. Not only has the DNA we collected degraded over the past couple million years, but its resulting RNA needs to be correctly spliced. We had to figure out, through much trial and error, the correct sequence of splices to create the proteins that manage the cells of the dinosaurs. Fortunately, the records on site were recovered in the late 1990s, and they gave us the foundation of our continuing research. From what I hear, Mr. Wortman Whitfield is here to talk to us, so I'll let him finish talking about how we made the dinosaurs. Wortman.
After that first successful DNA extraction, we still had to get a living animal out of it. Most of the time, we weren't even successful. Though neither were the scientists at the first park. At first, we followed in their steps and kept improving the methods where we could. Individual proteins do not make a dinosaur. However, thankfully, in Site B documents there were experiments that outlined to determine the function of many of the proteins we were seeing translated the gene sequences we were finding. Comparisons of our dinosaurs' genetic code to the codes of the first various species, minus the frog splices, showed us that indeed, we were very close on many occasions to complete DNA strands. We still needed to splice in other species for the others. This time, we used avian DNA, or ornithosyncynoids. Since many lineages of dinosaurs evolved into present-day birds, we also used saccharoids, received reptilian DNA if necessary. Mr. Hammond has labs around the world working on genetically mapping all the modern-day birds and reptiles. Each time we extract DNA, the database of modern and Mesozoic species we can compare to has grown, and we get closer and closer to the real thing. Once we lay what we assume is viable DNA, it is injected in vitro into female ostriches. The ostrich then lays a fertilized egg with our dinosaur's DNA in the zygote.
Wait, does that mean there's actually dinosaurs here? Do we get to see them?
Yeah.
No way.
Yeah.
Way.
Sweet.
I know. Now, hey.
Did you guys not read the pamphlets we sent you?
Yeah.
No, not at all. Didn't even look at it. How'd you get
Yeah, we
here?
don't do that.
I don't know.
Did you guys not see the big
Nope.
brontosaur in the land?
No.
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 Gone.
You guys took the crappy way in.
Yeah, really.
But I will say, the proof is out there of all this awesome work that we've got going. The protein in the eggs act as analog chaperone proteins for our dinosaurs' proteins. They help fold them into the correct shape where the natural chemical proteins of the peptides do not automatically fold the protein. As our animals have been growing, we have been taking samples of their cells and examining their structure. We hope to be able to begin manufacturing synthetic versions of these chaperone proteins within the next few years.
All right, and with that, thank you, Whitfield. Would you guys like to go see the park? The jeeps are ready.
Nah.
We don't care what you have to say. Would you guys like to go see
I'm
the
down.
park?
Let's go.
All right, let's
Wanna
go.
go?
Sure, let's go. I got my camera ready.
The dinosaurs got out! The dinosaurs got out! Dinosaurs are out, we gotta go. Let's go. Come on, shuffle a little faster here. Gotta go. Big ones, large ones, out of the pens. Let's go.
Wait, what?
What?
What's a
They're
dinosaur?
out?
Dinosaurs.
Which ones?
Like,
All of them, the ones with sharp teeth.
Like,
will
what's
out.
you let them out? Like,
What-
what's a dinosaur?
How did they get out? What about the fences?
Dude, don't ask questions, run. Just run. Dude, what are you doing?
But,
What
like,
are you?
I don't think this is a really severe situation.
I think we'll be okay.
Wait,
Are we safer
Like,
in the building?
Stairs.
Like, I don't
no,
see-
no. We need to get out of the park.
But like,
Why?
I don't take the stairs. Elevators.
Stairs.
Elevator.
I had
The
a,
elevator.
I bought the damn elevator. I'm
Just
using
take
one.
it, okay? It's okay.
Come on, everybody. Go. 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 Take my car.
Get in.
I don't run. Every time I touch
Hurry.
it, I don't run.
right there.
Somebody scream.
Ah! Oh my god! Ah! Like a good neighbor, big bonus now.
I got that down here.
You are a dinosaur. You are a dinosaur. O-L-D-M-A-N. You're just an old man hitting on me. What? You need a CAT scan.
I'm done for the night. I can't do anymore.
Oh my god, that was rough.
Yeah, buddy.
I'll call you guys later.
Yeah, buddy.
Fuck that. That's stupid.
All right.
No, actually.